So let me start by thanking Debashish for and Prabal for inviting me to this talk. Am I audible? Oh, awesome, awesome, great. Thank you. The last talk was by a professed biologist. This time it's again, we're getting worse. This talk is by an ex-clinician who looks at interesting biology in clinical problems. So bear with me as I pour more biology onto you. But I think I'll try to flesh out a few interesting morphological principles as we go along. Morphology is, of course, a fancier word for a more ominous term called anatomy. So anyway, we'll get there. I do look at cancer for a living, and so I'll talk about which cancers and why and so on in just a bit. This is, of course, a mass of cancer cells which has been embedded in a three-dimensional environment, which is similar to the kind of environment the cancer cells find themselves in within the body. And just to sort of segue into the kind of stuff that I'll be talking about as to how these cancer cells start breaking out from their confines and start interacting with their surrounding substances. And I'll of course elaborate on these substances as we go along. Right. So the first principle, or the first sort of common property, and that is that what makes a nose and what makes the liver a liver, or rather what makes a nose different from a liver and vice versa. And if one poses this question, one can answer this on many different levels, but one way of putting this across is when one looks at cells, one sees that cells within the nose are arranged very differently from the way they are arranged within the liver. And that's pretty much a sense of the reality one gets when one has to speak to a surgeon or a pathologist in the clinic. And so when a pathologist is looking under a microscope, what she's essentially doing is identifying a certain cellular pattern, a certain cellular arrangement. And using this sense of arrangement, using this strong, intuitively built sense of arrangement of cells, she or he is saying, okay, this looks like a sample of liver to me, or this looks like the kidney to me, and so on. And based on, again, on this arrangement of cells, this stereotypical architecture, which is very common, which makes the arrangement of cells in the nose different from the arrangement of cells in the liver, the pathologist can go beyond just saying, okay, this looks like a nose or liver, and move on to say, this looks like a normal nose, this looks like a normal liver or an abnormal liver, and so on. So the arrangement of cells, what developmental biologists call patterns, cellular patterns, is pretty much what decides what a particular bunch of cells within our body is going on to become. And that's what makes one organ very distinct from another organ. And it's precisely these patterns, these architectures, that make organs and tissues very unique with respect to each other. It's these patterns that are violated, that are subverted when you have cancer. Okay. So cancer can be defined, again, on many levels, depending on who is defining and describing cancer. But one of the ways of defining cancer is the loss of that archetypical pattern that makes that tissue or organ unique with respect to each other. Okay. All right. So what I have here are cells which have been arranged, or rather cells which have been cultured within a particular three-dimensional environment. The black here is not space, it's what is known as extracellular matrix. So what are tissues made of? In the previous image, what we are looking at here are basically stained cells. We're looking at cells which have been stained with specific dyes that pathologists typically put onto cells so as to mark their nuclei, as well as mark their cytoplasm. So you have these two components of cells, the nucleus and cytoplasm. So they've been marked with two different colors, and that allows pathologists to look at cells and therefore make out the cells and see how the cells are arranged, right? So you see that these organs, the kidney and the liver, have specific patterns, as I mentioned, the archetypical patterns that make them unique. Tissues and organs are not just made of cells. When you remove all the cells from an organ, you still have a structure, in your hand. And that sort of structure, or the house, or the scaffolding, or whatever you might want to call it, is known as the extracellular matrix. This extracellular matrix is a bunch of proteins, carbohydrates and lipids, or conjugated with each other. And this sort of composite has certain very interesting properties, interesting chemical properties, interesting mechanical properties. And these properties, again, make that an organ, the extracellular matrix of the liver, is very distinct from that of the kidney. And just the way cells and cellular patterns make these organs distinct from each other, even their extracellular matrix, the part that's outside the cells, also make them very unique with respect to each other. So what people frequently do, what experimentalists do frequently, is that they are able to take these cells from a particular organ and put it within the extracellular matrix of that organ under a microscope and study the processes associated with that organ, development, disease, call it what you will. So these are breast cells. These are there's a term called epithelial cells. So they are breast epithelial cells. By epithelial, typically mean cells of a particular shape. And these are cells that have a cuboidal shape when you put them within the matrix that is typically associated with the breast organ. Okay, so we've taken breast cells and put them within the matrix that is unique to the breast. And what happens when we do that? When we do that, they start assuming the archetypical pattern that you would typically see these cells to assume within the organ of interest, which is breast. So in other words, how are breast cells arranged within the organ? Breast cells form these long ducts, hollow ducts, and end up within these they form these cavitations at the end of these ducts. All these ducts, of course, unify and come on to eventually come out to the nipple. Now, at the end of these ducts are these sort of cavitations, the bulbous ends, and these ends have these cells surrounding them with a hollow in the middle. Hollow in morphological terms is called a lumen. So these cells, when given the right kind of cues, the right kind of hormonal cues, which again are typical to what one would see when one is lactating, they would start secreting milk protein, and this milk protein is therefore now secreted into this hollow area, and in the organ, of course, it will flow out through the ducts and come to the baby. Right? So, but even in culture, when we take these breast epithelial cells and we put them in the matrix, they form this particular stereotypical pattern, similar to what you would see in the organ. And if we were to give it the right kind of cues, these cells would also start secreting milk proteins. Excuse me. So there you go. So we are able to therefore recreate the pattern in a three-dimensional space under a microscope, and therefore we can study breast physiology. Physiology, of course, means normal functions, and we can even study breast structure. Anatomical questions, morphological questions can be asked within this system. Now, what happens when you get cancer? Now, these cells, when they become transformed, the term is transformation. In other words, there are certain changes that occur within the DNA of these cells. They start dividing, right? And I describe cancer as a departure from the stereotypical pattern. So you can now see that this particular pattern in which cells formed a hollow around them is somewhat now altered. So what we are seeing are basically digital renderings, three-dimensional rendering, or 2D renderings of three-dimensional structures. So you can see that these cells are no longer now hollow, have a hollow inside. They have filled their inner sort of cavitation and have formed a ball of cells. Now, these are breast cancer cells, which are not of a very aggressive nature. So all they do is they divide a bit fast, and then they form these kind of rounded spherical structures. What happens if the cancer gets worse, if it gets more and more aggressive, as it's called? So these breast cancer cells now start not just dividing, but also migrating. And you can imagine the video that was playing in the beginning, that those two were breast cancer cells. Now they start dividing, they start streaming, they start moving into their surrounding matrix. They start eating the matrix around them. They start remodeling the matrix around them, and they form these branches, and through these these branches of cells start moving through the matrix and go here and there. So that's your narrative of cancer. Cancer represents this particular trajectory, and cancer therefore has often been described as a loss in the pattern, as a loss in the architecture that goes on to give rise to tissues. But one can therefore also turn the question around its head and ask, if that's the case, can one also envision cancer as a complex process by which a set of interactions, a set of, say, cellular interactions that give rise to one particular pattern are now replaced by a new set of interactions, a new set of interactions between the cells and the surroundings that lead to newer patterns that emerge, newer possibilities that arise from the cells and from the DNA that codes for the genes that code for the proteins and so on. All right? So this is a slightly more complex version of understanding of cancer. And I'll build an even more complex version as we go along. So to understand the process of seeking new rules and understanding these new rules of interactions, one can again go back to the original mother subject, which is developmental biology. And one can therefore ask, how does form change along embryological time scale? So if one wants to look at cellular interactions, cell environment interactions, by environment I mean the matrix in this case and so on, how does that happen within developing embryos? Okay, that itself can give you some clues as to how things unfold. And of course, one can extend this to a broader question as to how do these rules of forms also changing along evolutionary timescales? Because eventually, not just do you have development, but the way you would want to see how organisms change over evolutionary timescales or rather organisms evolve over evolutionary timescales, and new species are born through new modes and new grammar of development that evolves over time. Okay. So to try and handle these questions, there have been multiple approaches by people with very different backgrounds and strengths. And one of the most I would say, a genetic strong narrative for a very long time came from this brilliant developmental biologist Eric Davidson at Caltech, and of course a lot of people working with him. And this sort of presaged the concept of what at that time was maybe in the 1670s was somewhat novel, but now is of course a very common term called gene regulatory networks. The idea that genes are linked to each other and their expressions are linked to each other in the form of these very complex networks, and these networks and how these genes are arranged and what genes are arranged in certain particular ways, and their interactions give rise to specific cell states, and probably with the idea that specific cell states give rise to specific tissue states. So again, going back to the question of how did the nose become the nose and the liver becomes the liver, is because there are a certain network of genes that are upstream the formation of the nose, and there are a set of genes that are upstream the formation of the liver, and they are distinct with respect to each other. Sounds very okayish right now, but was at that time, it's one thing to say it and difficult, it's a challenging thing to prove it. And so a lot of very, very good molecular genetics, developmental genetics was used to try and understand this process, and a lot of good work went into it. So first question, sort of one approach to solving that first question I mentioned, what are the rules by which embryonic development could be tackled? And what about the second question? How does form change? How do these rules change over evolutionary time? So if one uncovers these gene regulatory networks, can one use these principles and can one use this grammar to uncover how different organisms develop? And so yes, there was work done along this school using this approach, where again very subtle changes in these networks were also found to give rise to the same developmental stage in related organisms. Okay, so within echinoderms, which are a certain class of complex multicellular organisms, one could see that the gene regulatory network, the set of genes that give rise to a particular cell state, had some distinct, slightly subtle but distinct features from the set of genes that were giving rise to the same cell but in a related, evolutionarily related echinoderm species. Okay, so clear. Okay, fine. So we can use gene regulatory networks as a good tool, as a good conceptual tool to understand how certain cell states come about in development. We can also use this as a good conceptual tool to understand how development can ever so slightly get altered when it comes to related species. So it allows us to not just understand the development of organism A, but also organism A prime. All right. Fine. But one of the interesting sort of asides from this, or rather, a complementary approach to this was to say, okay, fine, we're looking at these genes. A lot of these genes are encoding in the gene regulatory networks. A lot of these genes encoded transcription factors. What are transcription factors? These are proteins that reside for a long time of their life within the nucleus and sort of link to each other, turn on each other's gene expression or regulate each other's gene expressions by binding to DNA. But if I were to try and understand how a cartilage cell differs from a bone cell, for example, or how the development of cartilage differs from that of bone, one wants to look at certain morphological principles and look at proteins that actually go on to mediate what makes these tissues different. So what are some of these proteins, and how are some of the players involved actually in carving out the physical and chemical properties of tissues and make them distinct with respect to each other? The transcription factors did not give that set of information. So this required a complementary approach. So a lot of work went in terms of trying to understand how a lot of players work outside cells. So these are proteins that get secreted outside cells or are present on cell surfaces that bring about processes such as cell adhesion, intracellular adhesion, that bring about adhesion of cells to the matrix and so on, and try to look at the evolutionary importance of these kind of proteins that are actually mediating certain specific morphogenetic processes. Now, these set of proteins, which are either present on cells, so can act as, say, a cellular glue, bringing a bunch of cells together. If they're expressed, you would have 5 cells separated, and now expressed, these cells would sort of come together. So these proteins and these players were seen to work across cellular scale. So rather than being in the nucleus, now they would come out, and as they come out, by dint of their being secreted, by dint of their being present on the cellular surfaces, you would start seeing interesting developmental processes emerging. So in this way, one could imagine how, in one way, unicellular organisms could have evolved to give rise to multicellular species and so on, because they were able to bring cells together, pattern cells in different ways by modulating the adhesion of cells, by modulating the presence or absence of cells, of cells at certain locations, and so on and so forth. So this is a sort of a complementary approach to looking at development and evolution. And again, to a great extent, these processes and these players and these specific interactions outside the cell were able to explain certain interesting developmental features. One example, for example, one case, for example, is limb development. So in limb development, the way our limbs develop can be sort of reduced to, in developmental biological terms, to understanding how the skeleton develops of the limb. And the way skeleton develops of the limb again can be reduced to understanding how fingers are sized in space. So this becomes more of a sizing and spacing problem as to how one can have a set of events occurring simultaneously spaced apart in a particular regular fashion. And a lot of very interesting work has gone on to try and unravel this particular pattern that happens within living embryos as they develop these limbs and as they develop these what are called digits. So it's a problem of digit patterning. And since the 1970s, Hans Meinhardt and Gierer and going on, others have tried to look at this problem from the point of view of some very interesting chemical theoretical ideas that were proposed by Alan Turing. So again, as per Meinhardt's framework, if one could have certain activators of this particular patterning event or formation event and inhibitors of the same event, and if these could lay down some kind of a chemical map in which you had the activator concentrations at a higher level in certain areas, followed by inhibitor levels sort of peaking in other areas, one could give rise to a certain space, a pattern in space, which would give rise to some kind of a chemical sort of a chemical map, according to which now certain development events can take place in a spaced apart fashion. And there was a lot of work that was done to try and unravel whether there are such chemical mediators that give rise to this kind of pattern. James Sharp's group came up with a very interesting sort of interaction network, which included 3 molecules. The identities are irrelevant. There was work done from our group some time back, which also looked at this event in terms of certain molecules that would again diffuse and that could bind to certain receptors in some particular interactive manner, with certain experiments showing that this was indeed taking place and so on. So chemical maps laid down by what are known as morphogens. So the term is morphogens, diffusive molecules that can pattern their concentrations in space based on certain reaction and their diffusivity can give rise to tissue patterning. So this is the other approach. Now, so on one hand, one would have transcription factors and networks of transcription factors giving rise to certain cell states. On the other hand, outside the cell, you would have chemicals that are able to mediate processes or lay down maps based on their concentration. But there's something else that is very interestingly happening in the middle, and that has been missed in both these approaches, and that's critical to a lot of events that now we are up against and are hard pressed to explain. One example is this comes from Javier Trepad's lab at IBEC in Barcelona, where what Trepad has done is that he's taken these bunch of cancer cells, same cancer cells that I showed on the right hand side, the cells that, the aggressive cancer cells that sort of sample their environment. And what he does is that he tries to look at their ability to wet a particular substratum. So what he's done is that he's sort of created a space for the cells to sit on, and the rest of the surface the cells cannot sit on. And so this sort of like stenciled a certain space. He's put the cells in there, and then he takes off the stencil. So now the cells can spread. And so this one can think of this like a droplet or water spreading or liquid spreading on a surface. Now the interesting thing is it starts spreading, but after a while it starts wetting the surface around it. After a while it starts de-wetting it. And this is a very interesting thing as to how it starts de-wetting the surface. And what comes out, this is a beautiful paper in Nature Physics, and what seems to be very critical in determining this wetting followed by de-wetting is the fact that one cannot explain this purely by using the 2 typical forces used in wetting phenomena, which is, in this case, the agent and substrate energy and the interagent sort of energies, or in this case one would think of this as the cell substrate energy and the cell energy. What the process seems to involve the internal workings of the cells, which is the cellular cytoskeleton. So in other words, what seems to be happening is that as the cells start spreading, at some point of time, there is a very strong actin buildup. And the actin builds up, the cytoskeleton builds up, and this actin is linked to all the cells. The whole patch works as a single mass. And all of this actin starts giving it a sort of a contractile force. And this contractile force that builds up at a particular spatial length beyond which it starts sort of ripping off. And now it starts goes from a monolayer to a 3D. And so it starts sort of de-wetting itself. Okay? So this kind of process cannot just be explained by the cellular adhesion, the adhesion that is between the cells that I was talking about in the chemical approach or in the early approach. It cannot be explained simply by adhesion of cells to the substratum or the cell-cell adhesion. It requires It strongly requires an active cytoskeleton that builds up within it to understand what's going on. And the way they showed it is that if you are able to sort of decouple the actin and myosin by using certain inhibitors or using other inhibitors of signaling, such as what is called the ROCK inhibitor, which again decouples the actin buildup, one starts seeing delays or impairment of the de-wetting phenomenon. Okay? So in other words, not just chemical processes, genetic processes, but also very important cytoskeletal active processes are all working together to give rise to the architecture, to give rise to our anatomies in normal and abnormal states. So this is the next sort of small morphological principle that I sort of flesh out from here. Yes, Shakuntala. Okay. Okay. While we have not gone there, and neither has Xavier, but I will show you some processes of at least wetting, and I'll show you that that process in a pathologically relevant context towards the end of my talk. Okay? Good question. So the cancers that we work in our laboratory are breast and ovarian cancers. And the reason why we do that is also, there's a reason why we study these two specific cancers. The first one is basically a cartoon depiction almost of that first video that I showed you of cells coming out. And of course, the second image of the breast cells within their microenvironment. You have basically, you have these cells that are arranged in a stereotypical manner, and this is the extracellular matrix shown in the form of these orange lines, which is basically one of the most abundant proteins in our body, the collagen. And then cells sort of start filling up, then they at some point of time they break through their confines, they start entering into their surrounding extracellular matrix, and then they basically seek blood vessels, and they get into the blood and start spreading into other parts of the body. Typical narrative of cancer. In ovarian cancer, something else goes on entirely. So breast cancer, the way breast cancer kills is not because it renders the breast dysfunctional, because the breast is in that sense not a vital organ for the existence of the individual, right? So the way breast cancer cells kill is that these cells enter the blood vessels, and from there they go to other parts of the body, parts like the liver, which is important for metabolism, most of the metabolic functions of our body, right? It goes to the brain, it goes to the bone marrow where your blood cells are being made. And in this process, it starts taking out all the vital organs of the body, and that's how it kills. So metastasis, this process is called metastasis. It's metastasis that kills in case of breast cancer. So ovarian cancer is a very, very odd disease in that sense that it does not seem to metastasize to different parts of the body. The ovarian cancer develops within a hollow cavity of the individual, of the woman, known as the peritoneum. The peritoneum, of course, exists in men as well, but in women, it's a large enough cavity which is present in the abdomen. And the way the peritoneum is organized is that it's sort of the negation of all the area that is not filled up by other organs. So what are the organs in our abdomen? We have the liver, we have the intestines, and the stomach, the intestines, and so on, right? So all the stuff, all the area that is not occupied by these organs is the peritoneum. And it's lined by a lining. So basically that lining is the exterior lining of all the organs and some space. Okay, and it's filled with fluid. So that this peritoneal cavity is the battlefield for ovarian cancer. Why? Because like all the other organs I described, the ovary is also present similarly at the edge of this cavity. Okay, so what happens is that if you imagine this to be the boundary of the ovary and the peritoneal cavity, what you get is that cancer cells from the ovary start getting shedded into this fluid-filled cavity. Once they get into the fluid-filled cavity, they start aggregating and forming structures known as clusters or spheroids. And these structures called spheroids and so on now start floating around in this cavity and then start homing on to certain areas of this cavity where they colonize. The term is, it's a very apt metaphor in that sense. It colonizes that area and starts sort of spreading there. ovarian cancer in that sense is very interesting. It doesn't seem to go too much outside this cavity, because here itself lies the intestine, and as it reaches the intestine, that's where it's sort of it richly homes onto and starts colonizing. As it reaches the intestine, it starts sort of growing on the intestine and starts squeezing the intestine. And because of this, you get what is called intestinal obstruction. There's loss of appetite, and that leads to deranged metabolism, and eventually sort of the weakening of the individual and eventual death. So that's the way ovarian cancer kills. So the metastasis is somewhat local, and it is mediated by these structures known as spheroids. So the idea is, how do we understand metastasis in general? So one can again break down the process of metastasis into a few important sort of theaters. So the first one is, of course, early migration from the primary focus, from the first point where the cancer arises. What happens very locally? How does the local architecture break, and how do cells move out? The second one is where it sort of enters into a particular fluid environment. One can think of it as the breaking of breast cancer cells into blood vessels, or the breaking of ovarian cancer cells into the peritoneal cavity. The third process is migration within these fluid environments. As these cells move through the fluid environments, how do they survive these environments, and so on. And the fourth process is, of course, colonization. Once they've reached where they want to reach, or where they are happy to now settle down, how do they colonize, how do they grow there, and so on. So these are sort of four paradigms, if you will, of metastasis, but one can break it down to. So the story that I'll briefly talk about concerns this particular process, because I think of it as a very interesting morphological problem. Why is it important to study spheroids? The reason is because, again, the spheroids pose a very interesting problem. Typically, one sees cancer cells or cancer as breaking down of multicellularity. In fact, fancifully, people, I would say loosely metaphorize cancer as breaking down of multicellularity, where cells now become almost lone wolves and start doing what they're doing. What we see here now is some kind of a recapitulation of cellular complexity. So when I spoke about that bursting of the primary focus and cells start coming out, they seem to lose their intercellular adhesion. Here, cells seem to gain intercellular adhesion. So as I'll go along, I'll show you how there are certain interesting principles of organization that cancer seem to gain. So just going back to the idea of, are there new rules of interaction that cancer cells seem to recapitulate? And that's the kind of argument that I'll try to build up. Why is it important to study spheroids? They are the consequences. They lead to metastasis. They lead to what is called the recurrence of the disease. The reason why the disease seems to come back again and again, because you're taking out the ovary, which is already tumorous, but these spheroids have gone into the peritoneal cavity and are hiding there. And it's very difficult to take them out, because they are, of course, microscopic structures. So the surgeon can't take out spheroids and so on. These cells often become resistant to any kind of drug that you put them on. So the cells within the spheroids seem to be resistant. So that makes them extremely dangerous things. Of course, these spheroids are the ones that are the mediators of colonization. They are the ones that actively go to the tissues and start colonizing, and of course cause some of the downstream events associated with colonization, which I described. Yes. Oh, I'll talk about heterogeneity. It's actually a problem of heterogeneity. So there you go. You predicted the next slide. So when we looked at spheroids from patients, so what we got was we got the fluid from patients who were suffering from ovarian cancer. And the idea at that time was I was interested in trying to study how cancer spread within the body. It was a simple problem. Wanted to get basically cells from blood. This was a breast cancer problem I was pursuing. Completely failed there for reasons I can get into later. And then somebody said, why don't you look at ovarian cancer? It doesn't come to blood, but it comes into the peritoneal environment, and we frequently tap out the fluid and we throw it away. Eventually, we don't even study it. It's just to relieve the symptoms, because this fluid accumulates in the abdomen and starts giving pain to the patient. the idea is to remove this fluid out and give relief to the patient, and otherwise we have no use for it. I said, great, let's take this fluid, let's put it on a microscope, let's study it. When we looked at these spheroids under a microscope, we were intrigued by something very interesting, and that's something that Sandhya mentioned, and that is that a lot of these structures, a lot of these multicellular structures were either solid, in the sense they were made of a bunch of cells come together, but also they seem to have cavitations within them. They seem to have a lumen within them. Now, if you remember my second slide, I was talking about the lumen in a normal context. It was the normal breast cells which had this lumen and cells surrounding it, right? And the loss of lumen was associated, the loss of architecture that one sees in breast cancer. It's of course a breast cancer story. Here we see a sort of recapitulation of a normal architecture in cancer cells. Okay, so anyway, so we found that there were these kind of spheroids and there were these kind of solid cell-filled spheroids. And the question was, why do we see these two spheroids? Do they sort of, are they stable entities? Do they transition between each other? Is there reverse transition? And so a bunch of morphological questions can therefore be asked, and we have, of course, other tools by which we can investigate the formation of these structures, because when we look at it in patient spheroids, of course, very hard to see how they formed. We are getting them de novo, so we have to develop some models to actually witness their formation. Yes, Rup. Oh, great. The way one can figure this out is through multiple ways. One is, of course, given that they are not very big, they don't have a lot of cells within them, about 30 to 80 cells. With phase contrast microscopy, one can actually visualize both these sides, because you can see the thinning on both sides. Of course, then one can resort to confocal microscopy, scanning electron microscopy in broken spheroids, and see a very smooth contour inside. So a good way of asking is it a cavitation versus a lumen? A lot of people do fuss about this, and I'll show you why we think it's a lumen and not merely just a hollow that has developed in the middle. Okay? But yeah, good point. So these are, of course, again, scanning electron micrographs of cells that we have used to form the spheroids. This is what is a solid spheroid. So just as an elaboration of what I answered, one can use confocal microscopy to actually look at the insides, and one can see that the inside of this is filled with cells. And interestingly, these are the spheroids that these over time mature into. So one can now see that when one looks at these spheroids, these are the ones that have a hollow lumen inside them. One also sees that they seem to be covered with some kind of a coat, because one can no longer see the furrows in between the cells that one can very clearly see in this image. So they seem to be covered with something very interesting. And then when one takes these coated spheroids and puts them back on a substratum, what one sees is that they almost seem to be breaking this coat, and then eventually they spread out and spread onto the substrate. All right. So this, in some sense, also represents a narrative of what we see inside the patient's abdomen, in the sense that there is an adhesion that eventually takes place on the sites of colonization, and one can see these two spheroids. This, of course, moves into this gradually under the microscope as we passage them, or as we view them. And so, again, one asked, what is that coat, and we asked what is that coat doing, and of what use is it? And we very quickly again figured out that this coat that we could see on those sort of second type of spheroids, now we're talking about the heterogeneity, on the second type of spheroids which had this coat and which had a hollow inside it, had certain proteins. The names are not very important, but it's collagen and laminin. These are proteins that are, again, archetypical of the extracellular matrix that one sees in those breast hollow structures in normal morphogenetic examples. Okay, so again, going back, it's not just the cellular pattern, it's also the type of extracellular matrix that one sees in normal, and I'll now generalize this from breast to normal glandular tissues. Glandular means glands, sweat gland, sebaceous glands, hair follicles, prostate, breast, so on. All of these have a particular pattern of cells surrounding a hollow lumen, surrounded in turn by a specific type of extracellular matrix called the basement membrane. Okay, the name, for whatever reasons, forms the basal side of the cells and so on. So this particular basement membrane was also arising in these ovarian cancer spheroids. So these are again cancer cells that typically break this structure and go out. They are known to degrade the basement membrane. In case of this particular fluid microenvironment, in this fluid microenvironment, they have regained these principles of morphogenesis that are typically lost during cancer or during carcinogenesis. Okay. All right. I'll skip this slide for lack of time. Some interesting properties. Again, fun experiments and some interesting properties are these stable structures. Do they release cells or do they gain cells? Do they mix with other spheroids? Funky experiments to do in lab, and so we did them because we could do them. So we looked at these solid spheroids and we sort of bombarded them with single cells of a different fluorescence nature so as to be able to track the identities. And we quickly saw that these solid spheroids were more sort of morphologically unstable entities relative to these hollow membrane covered spheroids. The solid spheroids seem to accept new cells when we would give them, when they would be present within their surroundings. They would also very interestingly show coalescence with other solid spheroids. So if you were to have many solid spheroids close together, they would coalesce to form a single spheroid. And these features were absent in the hollow spheroids which had the membrane around them. Those would not accept new cells, those would not coalesce with other such hollow spheroids. Okay? And what, that's fine, but what is that membrane doing? Yes. If this, yes. Yes. You would only see macrophages which are policing the abdomen. So the peritoneal cavity, one of the major functions of the peritoneal cavities, gives a great place where macrophages can just filter out from the blood, move around, check the abdomen, which is of course a great place chock full of bacteria, see that there is no infection going on and get back into the lymph and flow away. So you see very few macrophages in the peritoneal fluid of normal people.
The reason I'm asking, it's very interesting that you see this mixing, but at what density are these present? That is it even meaningful?
Oh, they are the abdomen, the what is called the ascites fluid of ovarian cancer patients is full of these cells. It's full of these cells and spheroids. So we see that it's the spheroids which are mostly capable of metastasis, not the single cells. But single cells, as you can see, the single cells can be party to formation of the spheroids. So in that sense, there are dynamical exchanges which we seem to have uncovered at high densities, no less, but that can happen.
Is this the reason why ovarian cancers typically are much more aggressive than breast cancer?
Yeah. And I'll show you a few features of why that is so as well. But coming back to the slightly more dev bio experiments, again, what is the membrane doing? So we see that the membrane is present, we see that the lumen is present. Are the two coincidental or are the two associated? I think the video stopped. But anyway, the left video was supposed to show that when we remove this membrane by enzymatic removal, the lumen is lost. Okay, and when we wash away that enzyme that removes the membrane, the lumen is regained. So they now start regaining the lumen. So in other words, the basement membrane, that matrix, that external coat is associated with this lumen and with the structure. So all of these processes seem to be linked together. How? So how is this linkage happening? So to understand this, again, one goes back to the principles of intercellular junctions. How are cells linked together to form that archetypical architecture? And one needs to look at a series of sort of junctional complexes that form between cells. There are several names and there are several types of them, but let's again stick to nameless principles. Okay, so what we saw was that the basement membrane coat, the coat was increasing localization of one particular molecule called ARP23. Okay, at the intercellular junction. So we could see that as the solid structures were becoming hollow structures, there was strong localization of this particular actin stabilizer. So now again, coming back to that idea that you need the cytoskeletal elements to also give rise to certain morphogenetic principles becomes relevant. They would be strongly present at the edges, and if you were to remove the basement membrane, you would again have a loss of this particular actin stabilizer ARP23 at the intercellular junctions. And what happens when, of course, you don't have this actin stabilizer? So using, again, pharmacological approaches, we were able to show that when you remove this particular ARP23 molecule or inhibited its function, what you got was that the hollow structures now became solid structures and in fact disaggregated as well. So the actin stabilization is instrumental to the formation and progression of cells from a unicellular state to a solid state to a hollow state. And moreover, what was also happening is that, well, this is just a control experiment, that they are indeed forming these very strong actin complexes within the spheroids. Yes. What the ARP23 was doing is that it was also putting on in between the cells this particular cadherin molecule, which is involved in actually acting as the glue. So again, going back to my morphological principles, the thing that is present outside the cells is your cadherin. Well, not outside the cells, it's present in the cell surface. The thing that is present inside the cell, ARP23, are both now coming together. So all these principles are sort of coming together to allow us to understand morphogenesis. So ARP23 was also important in bringing these cadherins between the cells, and when it gives rise to these kind of spheroids. ARP23 was also involved in putting together the localization of another molecule called ezrin, which is again an actin stabilizer. It allows the actin now to get linked to the inner side of the cell membrane. As well as ARP23 was also bringing the localization of that basement membrane matrix on between the cells as well as outside the cells. So in other words, you had some kind of a positive induction between the basement membrane coat and processes that allow the cells to link together within the spheroids. Okay, so again, why do we need these hollow spheroids? Again, going back to a question that Sandhya asked. So we looked at these solid spheroids and these hollow spheroids by now it should play. We put these hollow spheroids and solid spheroids within these microfluidic tunnels, and the idea for doing so is because these tunnels are frequently seen, of such sizes, are frequently seen within the peritoneal cavity. And as I was mentioning, the macrophages, these cells which are immune cells, they police the abdomen and they go out through these sort of tunnels into the lymph vessels or the blood vessels after having policed. We saw that the solid spheroids, as they were coming out, were forming more deformed structures. You can see that the aspect ratio is altered in these solid spheroids, whereas the hollow spheroids were able to retain their aspect ratio even after they were being deformed. So we are deliberately deforming these structures, and we are seeing that they are able to recover their state, whereas the solid spheroids were not able to recover the state, so much so that we also saw that there was a greater level or greater proportion of disintegration of these solid spheroids with respect to the hollow spheroids. So now we were able to sort of distinguish the functionalities, not just that they're looking different, they're also behaving in certain distinct ways under certain challenges that we provide them with in terms of their mechanical behaviors as well. Okay, so that's great. Now we're looking at these hollow spheroids, and when we were looking at these hollow spheroids, we saw that they were not stable hollow structures. They're not like balloons that we have filled with, gas or water, and they're sort of stable entities. They also seem to be oscillating in their diameter with time. And this was something that we are working with Tapa Bhattacharya's group at NCBS, where we are trying to look at the external and internal diameters, and we see that there seems to be a particular frequency with which they seem to pulsate with time. So the idea, therefore, is perhaps that the mechanisms of morphogenesis, which one associates with in case of normal structures with being somewhat stable, may not be so stable in at least in case of these spheroids. And one of the at least candidate reasons why we may think that this is happening is because we do see that while the basement membrane increases ARP2/3, which increases the coherent localization, and it also increases the localization of ezrin, which brings the actin to that process. We see for some reasons that we are not able to understand that the inhibition of ezrin further increases the basement membrane localization. In other words, there may be an inhibitory loop here where ezrin seems to delocalize or decrease the localization of basement membrane onto the surface of these spheroids. And if this is the case, this sort of can, if this inductive kinetics and if this inhibitory kinetics are taking place at two different time scales, then one might be able to explain why this oscillation is taking place in terms of, they are trying to form these stable coherent complexes, but due to the ezrin buildup, and if it affects the basement membrane, then it breaks and it again comes down and again starts forming this process again. And so okay, I'm almost out of time. So I'll skip a few slides. Okay, so again going back to the idea of morphology and the idea of these hollow structures and why these hollow spheroids are more dangerous. So we showed in our experiments that hollow spheroids do not accept new cells into their midst. If we try to ask this question and ask this of other types of cells, are other types of cells also accepted into this structure? And to ask this question, we actually we expose them with monocytes and other immune cells. Now, these immune cells can actually kill these tumor cells, but because of their architecture, these hollow spheroids once again are able to evade the immune cells. So morphology might also give us a new principle by which cancer cells are able to evade immune cells. The hollow spheroids also seem to, for reasons again we don't know very clearly, accept less amounts of cancer drug within their structures. This could be because the way they have been organized, and there is a lot of literature that I think Masaki was talking about how the genome is organized. There's a lot of literature that goes on to show how the genome can be organized by tissue microenvironmental cues as well. And it could be that the way they have been shaped into these hollow structures is leading to expression of proteins that allow efflux of chemotherapeutic drugs from within these hollow spheroids. So the hollow spheroids exist for a reason. The heterogeneity exists for a reason, and they can actually give rise to certain protective survival responses for the cancer cells. Okay, I'll skip all of this. This is more clinical stuff, so this should be not relevant. I'll just end with a couple of slides. So, I was talking of all this time, I was talking more so about cancer cells and their immediate environment, which is a fluid environment. But if one looks at the process of colonization, one can ask certain other questions, certain more complicated questions, and look at cancer also as an outcome, not just of the behavior of cancer cells, but also of the defense that can be potentially mounted by the normal tissue that surrounds cancer cells. In other words, the colonization and the eventual sort of metastasis of cancer cells need not just be an outcome of what can be called the seed, which is the cancer cells, but also the health of the soil, which allows or disallows the seed to grow. Okay, so if one now wants to look at the agricultural properties of the soil, and therefore translating the metaphor, the physiological properties of these surfaces, one can then ask, what are the cell biological properties of these normal cells that would otherwise are present and doing their functions? How is the health of these cells regulating colonization? And that's where the idea of the environmental causes of cancer can come in and impinge upon the genetic causes of cancer, or not environmental causes, but environmental risk factors. So what are the risk factors? We know that aging and senescence, aged populations do get some of the cancers far more than younger populations. It's a very well-known data for that as well. And there are other risk factors. For example, diabetes is a big risk factor that is known to also affect and increase the risk for cancer incidence as well as cancer metastasis. So we are in the lab, these are ongoing projects. We're trying to look at how this process happens. And I'll just leave you with one set of interesting findings that we have. So these are the normal cells that are lining the peritoneal cavity, and we see, and these are of course the cancer spheroids, and we see that the normal cells are able to sort of prevent the cancer cell from attaching even when they are at high density and at some point of time at low density. So Shakuntala, we're talking about wetting process, right? So look at how this starts sort of wetting the surface. So at lower densities of these normal cells, the cancer cells are still able to sort of make space for themselves and start sort of spreading and growing out here. And these are the normal peritoneal cells that are, well, they're not normal now. They are the non-cancerous peritoneal cells which have been provided with a more diabetic-like environment. They've been exposed to the kind of fluid environment the diabetic cells encounter, and when that happens, you see that the cancer spheroids spreads onto the surface far more efficiently, far more faster, and so on, which again proves the point, well, which goes on to strengthen the point that the outcome of metastasis is both a function not just of the behaviors in cancer cells, which we all obsess with as cancer biologists, but also the physiological states of the microenvironment, which not just includes the extracellular matrix that I talked about, but also other normal cells which are continuously working around the cancer cells, probably trying to protect us from their onslaught, and under chronic diseased conditions not able to do so. So, okay, so I've kind of already stated the conclusion. Heterogeneity in morphology has consequences in cancer, and heterogeneous expressions can be harnessed to devise normal management strategies, because now if we know these novel morphogenetic rules by which we understand how spheroids form, now we can devise novel therapeutic strategies to take out those specific rules. Because in a lot of times what we do is we give our drugs against dividing cancer cells. In a lot of cases like these spheroids, they are extremely slow dividing cells. They don't divide as fast. So chemotherapy, the conventional chemotherapy often falls short in taking these spheroids out. So now we can use sort of morphogenetic mediators and players that affect morphogenetic rules to try and attack such processes and perhaps go towards newer forms of therapy. So acknowledgements, of course, to all the students who have worked with us, the clinicians who have been very generous to both work with us on the questions as well as the materials that we use in our laboratory. Ambarish and Proshenjit from the Center for Nanosciences for being fantastic collaborators on a bunch of processes, and of course funders for bravely understanding the kind of weird questions that we ask and supporting us for solving those. So I'll end here. Thank you.
Yeah, thank you, Ramray, for a very informative talk on cancer spheroid. And now session is open for questions.
Shakuntala. Ha. Ha, ha, ha. Ha, ha. A great question. We did try to ask this question, and we saw that there was not much of a proliferative difference when it came to these two spheroids. But just to add another tidbit onto this question, so if you have so many of these interesting properties of hollow spheroids, why do you need solid spheroids? And the answer to that came through when we took these spheroids and put them within mice peritonia, we found that the solid spheroids attach faster. So solid spheroids are able to adhere very fast. They can adhere to other cells, they can adhere to other spheroids, they can also adhere to surfaces, biological surfaces. So they are fast colonizers, but poor survivors, as shown by the microfluidics experiment. The hollow spheroids are elastic, resilient structures, are slow attachers, long survivors, which is why a lot of times when we open up the abdomen of the patient and look at these spheroids, many of them turn out to be the hollow spheroids, because the solid ones have either transitioned into hollow or they have settled down and are starting the process of adhesion. So in this way, cancer metastasis has evolved two strategies using heterogeneity of multicellular structures, one to grow fast and not survive long enough to become chemoresistant, and the other to not attach fast but mount a more chemoresistant more strong, survivalist form of invasion to further reaches of the peritoneum, perhaps. That part I don't know much, but I'm speculating on that. Roop, you had a question? Yes. So they the okay, why or how have they formed the lumen? Okay, what we know certainly is that they have regained polarity. They have put E-coherence and N-coherence, interestingly, on their intercellular, on their lateral membranes. Interestingly, ZO1 is not on the apical side, but it's on the basal side along with the basal membrane, which is again a very rare pattern that one sees in case of blastula, where again you have ZO1 on the outside rather than the inside to protect the inner cell mass from the uterine fluid environment. So they have gone on to form a unique sense of polarity, and they have the matrix on the surface. But it's, while doing so, it's not a very stable lumen. As you can see, it's an unstable lumen. So it forms it, breaks, forms it, breaks. And again, we think the reason why it does this is so that it can traverse through environments that have different variations of lumen or orifices as one sees in peritoneal cavity. Sure.